Welcome to the first presentation in the fourth webinar series presented by the International Adsorption Society. The IAS is an international organization dedicated to advancing adsorption as a solution to scientific and engineering challenges, human welfare challenges through the promotion of adsorption, education, and research. We thank everyone for attending the webinar today. The webinar series has been an immense success, and recordings of previous webinars continue to be viewed on YouTube. This is the first webinar of our fourth series, which we'll continue to have monthly throughout the year. We intend to continue with a variety of speakers from industry, academia, and other institutes, as well as PhD students and early career researchers. Announcements regarding the fourth series will be distributed through the IAS mailing list and the IAS Twitter feed. Today's webinar will be given by Professor Kim Jelfs of Imperial College London in the United Kingdom. I'm Nicholas Wilkins at Svante. Today's webinar will be moderated by David Donacci at Imperial College London. We're required to remind attendees and future viewers that the views expressed by the speaker, host, or other moderators are not necessarily those of the IAS or other institutions associated with those individuals. We ask that you consider joining the IAS as a regular member if you are not already. Our dues are minimal, only $20 per year, but support the publication of our flagship journal, Adsorption, contribute to travel grants, and workshop seed funding for IAS members and affiliated groups, as well as the organization of our triennial conference on the fundamentals of adsorption. Members also receive free access to IAS-supported materials, including our journal, as well as the adsorption database published by Springer Materials. Anyone can follow the IAS on Twitter at @intadsots for future updates regarding IAS events, webinars, and information about scientific meetings. Please help us expand our YouTube channel by liking this video and subscribing. The deadline for submitting abstracts for the first Mediterranean conference on porous materials, MedPor 23, has been extended to February 20th, 2023. MedPor 23 will take place in Crete, Greece, between the 17th and 19th of May, 2023. It is an excellent opportunity for scientists from all career stages to keep up with cutting-edge research in the field of porous materials and their applications. The conference includes many world-class scientists that will give keynotes and invited lectures, providing an important overview of the state of the art and future directions. For more information about the details of registration, please visit the website shown on the bottom of the screen. I'll now hand it over to David Donacci, who will moderate the Zoom Q&A.
Thank you, Nick. It's my pleasure to welcome Professor Kim Jelfs to the webinar series today. Kim is a professor of computational materials chemistry in the Department of Chemistry at Imperial College London. Her group specializes in the use of computer simulations to assist in the discovery of supramolecular materials. Kim completed her PhD in computational chemistry at University College London in 2010, studying the crystal growth of zeolitic materials. She worked as a postdoctoral researcher, conducting simulations in the groups of Professor Andy Cooper and Professor Matt Rosinski at the University of Liverpool, before beginning her independent research career at Imperial College in 2013. She was awarded a 2018 Royal Society of Chemistry Harrison Malodla Memorial Prize, the 2019 Philip Leverhulme Prize in Chemistry, and was also awarded the 2022 Blavatnik Awards Laureate in Chemistry, and she holds an ERC starting grant. Kim is an associate editor for Chemical Communications, and she joins us here today. During the webinar, questions can be submitted to the speaker either by the Q&A tab in Zoom or either in the chat of the YouTube live stream. Please leave any questions there for me to moderate, and questions will be answered at the end of the presentation. So with that, I'd like to welcome Kim again and pass over the presentation to her.
Thank you very much, both. So I'll just share my slides quickly. Yeah, David, if you just want to confirm that you can see
Yes,
that.
confirm that we can see and hear.
Perfect. Let me just move Zoom's toolbar out of the way so I can actually see my own slides. So yeah, thank you very much to both of you for the invitation. And it's a pleasure to talk to you today. I gave quite a broad title because obviously the IAS community has very broad interests. So I think what I've decided to do is to just sort of go into a little bit about various things that we're doing that are in the area of porous materials, not going into great depth into any one of them. So obviously if people want to follow up on any specific part later, they can do. But I thought to sort of show the overview of the different types of things we're doing in the area of porous materials. So more broadly, what we do within my group generally is to try to use computer simulations to assist in the discovery of supramolecular materials built from organic components. But really, this is quite centrally focused on porous materials. That is my own background. My PhD was in modelling of zeolites, so I've obviously continued that love of porous materials. And I'll be talking a little bit today, as I've mentioned, about porous molecular materials, which I started working on when I was a postdoc at the University of Liverpool with Andy Cooper, and have continued to be really interested in. I'll also talk about work in porous polymers, in particular modelling membranes of porous polymers. And I'll briefly touch as well on something. Generally, I try to avoid modelling metals, but I have a couple of things where we have been brave enough to also do the computer simulations, including metals, both in amorphous moths and in porous molecular materials that contain metals. So I'll touch upon both of those things today. But our essential themes, I guess, in particular of what we're interested in is really the structure prediction and how you can explore the chemical space to try to find interesting materials with great properties. And what I won't mention today is how we've extended lots of these structure and property prediction approaches also to organic electronics, but that is another theme of our research. So yeah, as I said, I'll start with porous molecules, probably because really I've been modelling those systems for the longest. Started getting interested when I was a postdoc. So a lot of our techniques that we apply to many different systems, we've got a bit further in terms of modelling the porous molecules. So very brief background into what porous molecular materials are. So unlike things like zeolites and moths, they haven't got that chemical bonding extending into 2 or 3 dimensions. Instead, they're made up from discrete molecules that in some way can pack in the solid state so as to give porosity, although they might also have interesting porosity in the solution state. But if we think about the solid state, there's 2 key ways that porous molecular materials can give you porosity. So at the top, I have extrinsic porosity, and this is where the molecules themselves pack inefficiently so that there are voids left between them. This is really quite a rare feature. Nature abhors a vacuum and generally will do a quite good job of managing to pack at quite high density so that you don't have voids. But there are some examples of this, and a real classic example is Dinene's compound, termed by Barra, or grandfather of zeolite chemistry, so to speak, as an organic zeolite. And that compound has those hourglass shape channels running along one dimension that can actually absorb some guests within them from very small gases. And then separately, you have an intrinsic porosity at the bottom, whereby the molecule itself has some sort of internal cavity or concave shape, such that you it has its own void space. There's various examples of this. What I'll be talking quite a bit about today are the imine cage systems, which in terms of purely organic systems are sort of one of the most popular types of chemistry for getting to these sort of highly symmetric cage type molecules. Generally speaking, these molecules typically have quite low porosity of the sort of hundreds of meters squared per gram. So nowhere near sort of some of the records of other types of extended network porous materials. But there is, I believe it's still the record holder as we speak, of a boronate ester cage from Michael Masterletz's group in Germany, which actually does have, well, a very large cavity, as you can see from the image, but also quite a high BET surface area of over 3,700 meters squared per gram. But in terms of not just the amount of porosity, in terms of the potential applications, I mean, these are still relatively newly researched, maybe in the last sort of 15 years or so. I've seen a real intensification of that. There are examples of this going back many, many decades. But so probably the most likely sort of application for these systems is in molecular separations and finding sort of unique application where their intrinsically modular molecular nature is useful, for instance, in improved solution processability. But because these cavities and the windows via which you access the cavities into the central cage are of the order of the same size as a lot of small organic molecules or small gases, that's why they obviously have potential for molecular separations. There's also examples of using them for sensing or detection. So, for instance, having a fluorescent moiety as part of the cage, but that fluorescence can be quenched when a particular guest is in the central cavity. For instance, if there was an explosive molecule that was in the central cavity, so that can be used for detection. In theory, they can also be used for catalysis, whereby that enclosed space is in some way altering the reactivity. And finally, it's not something that myself have done research in yet, but there's quite a lot of interest in use in porous liquids in general. So liquids that also have permanent porosity, sort of room temperature, and obviously potential processing advantages and so on from that. So the sort of concept was largely championed by Stuart James at the Queen's University Belfast, and cages are one of the first examples of being able to achieve this permanent porosity, so that they maintain their internal cavity, but also via functionalization of the cages, you're able to make them liquid at sort of room temperature or just above. So yeah, it's an interesting other application for these types of systems. So now we want to sort of walk you through how we try to assist in the discovery of these through computation. And really, I guess this is a battle towards getting predictive rather than rationalizing what's already been observed in the lab, being predictive and guiding our experimental colleagues as to which systems to synthesize. So first of all, if we explain the underlying structures of them, so much like many crystalline or crystalline porous materials will have some underlying topological net that you can relate their structure to. You have the same thing for the porous molecular materials, but obviously these are discrete molecular topologies, so it's platonic solids, polyhedra, this sort of thing. So in my example I've got here, imagine this is an octahedron, and in this case each of the yellow vertices that are 4 coordinated, you can imagine replacing with some 4 coordinated precursor. I can have a tetraaldehyde here, and then each of the purple edges I can replace with a diamine, and then you imagine reacting that together. This is an imine condensation reaction, and you have some hope of forming these sort of highly symmetric, beautiful structures as I've shown on the right. The imine condensation is quite a critical part of this. It's a type of chemistry called dynamic covalent chemistry, which is reversible. And that gives you the opportunity to not just form a polymer or some sort of other set of oligomers from the reaction, but the reversible chemistry may help you get towards these symmetric structures, particularly if they are the thermodynamic product of the reaction in the end, if there's enough time for sort of bond formation and breaking to get there. And that's one of the reasons that imine chemistry is by far the most popular type of chemistry by which these systems are made, because of this reversibility, but also other features of the imine chemistry that make the cages relatively stable and potentially porous, as I'll talk a bit more about in a moment. And I don't want to dwell on this too much, but something we did fairly early on in my group was to think through the different topologies that you could get if you're combining organic precursors that have different topicity or different number of reactive end groups. So at the top, if you combine a tritopic and ditopic linker, then in theory you could get a range of different topologies, which you get will be a factor of many different things: the geometry of the precursors, their flexibility, effects and so on. And we can see there's different families of topologies you could potentially access depending on your number of reactive end groups. And these ticks are just to show these are where there have been, or at least was at the time about 5 years ago, already existent literature reports of that type of topology system form. So topology also obviously doesn't necessarily mean the actual shape of the end system, but is the underlying connectivity. And of course, accessing different topologies potentially gives you access to different sort of size and shape of cavities, as well as different window sizes and shapes in terms of being able to do separations using them. And as you say, the sort of larger systems here, particularly in the middle, have only ever been reported in solution rather than being crystallized in the solid state. So the first thing that we would want to be able to do is to predict what the structure of any of these systems is going to be from the chemical reaction of the precursors. And so this is what we wanted to be able to do, is to be shown like this, just a ChemDraw image of the precursors, so just that 2D connectivity information, but to be able to go from that to being able to predict the structure. And there's different levels of that. So first of all, you have what is the topology that you're forming. I've just shown you that there can be various options from depending on the connectivity of the precursors. And you might think that maybe this should often be intuitively obvious, but it can be surprisingly subtle and easily easily get a different topology outcome from quite small changes of the precursors. So what I'm showing here is if you have a reaction with the same tritopic linker, the one sort of shown in grey, but you change the vertex only very slightly, then you get a totally different outcome. So on the left with the purple cyclopentane diamine, that reacts with a 4+6 reaction of 4 of the trialdehydes and 2 of the diamines to form the molecule shown there that is quite porous. It had a porosity BT surface area of about 1,300 meters squared per gram, which at the time when it was made was sort of a record for this class of material. But by contrast, if you just add in one extra CH2 group, as we see on the right-hand side with the sort of maroon-brown cyclohexane diamine, then in that case, you get an 8+12 reaction. So it's double the molecular mass. So very different outcome, as well as a different topology, it's a completely different shape. But worse than that, its properties are completely different. So in fact, that molecule ends up being completely non-porous, and also amorphous, and that's really unfortunate because the experimental postdoc that spent sort of a year and a half synthesizing that was hoping to get a really porous system, and it in fact wasn't. And this for us showed us the sort of value in doing modeling in advance that would tell you which topology you're going to get and also its properties, because you don't want to spend a year and a half going to the effort of doing all the sort of organic synthesis and characterization necessary here to find that it's not porous if that was your aim. And that sort of relates to this next point of what you want to be able to predict, which is shape persistence. So shape persistence I'm using here is the feature whereby even in the absence of solvent, the molecule maintains a permanent cavity. So it's porous. So as you get with the sort of balloons on the right-hand side, it's porous and open rather than the collapsed structures that you get on the left side that we say lack shape persistence. And shape persistence typically in these systems where we've actually done large screens of thousands of them is a really rare feature that maybe about 95% of the systems lack shape persistence. So you're sort of looking for the rarer sort of needles in the haystack of the shape persistent systems. And the lack of shape persistence is what explains the lack of porosity in the top right molecule that I just talked about. In the absence of solvent, each of those vertices folds inwards and removes your internal cavity. And largely it's due to too much flexibility in that system. And in fact, the solvate structure, which is the one that I'm showing at the top right, has within the crystal structure maybe something like 260 different DCM molecules that would be sitting somewhere within that cavity that would be scaffolding it to be open. But in the absence of that solvent, it loses all porosity. And the shape persistence is something relatively easy for us to predict generally computationally. We're just doing a short molecular dynamic simulations at a force field level, and that would predict whether the pore was open or not. And also more recently, we've built machine learning models that can give you that prediction in a few seconds as well. And then finally, if you did know the molecular structure, you also have the opportunity to do solid state packing, obviously quite key to knowing how its properties are going to be in the solid state. So we use crystal structure prediction techniques, in particular in collaboration with Graham Day at the University of Southampton in the UK. And these are techniques they often use on pharmaceutical organic molecules for polymorph prediction there, and apply them here to predict the low energy structures and therefore sort of the void network of these systems. And we can also do sort of annealing type simulations with molecular dynamics as well, if we wanted to predict how the system was going to pack amorphously, for instance, if you were casting a membrane. So we can do that, but it's non-trivial to do that. Like it may take weeks, months for a single system to do the solid state prediction. So as I'll come back to later, ideally we would want to avoid having to do that. And we certainly can't do solid state packing predictions for every system if we're trying to screen thousands of systems on a computer. But I did want to mention before I get onto the properties that each of those developments of predictive power took development and weren't trivial. And this paper from a few years back is an example of where we first put all of these elements together. So on the left-hand side, we did predictions of, in fact, we had a more complicated case where we had 3 different precursors. So we would predict how those 3 different precursors would combine at a molecular level. And because there's 3 precursors, it's actually more complicated because you could get social self-sorting where all 3 molecules form all 3 precursor molecules arrange into one supermolecule, or you can get 2 different 2-component systems instead, known as narcissistic self-sorting. And obviously those different outcomes give you totally different system, totally different porosity and so on. So we predicted the most promising systems that had the sort of hat type shape that I show at the top. And then our experimental colleagues went and synthesized the ones we predicted and got the hits in the lab. At that point, we then went with Graham Day and predicted the crystal structure packing before those doing crystallography worked on crystallizing the systems experimentally and getting the structures as well. And then we found from that we were able to pick up all the key sort of features that you would get in the end, as well in terms of the preferred packing modes, porosity, whether you get an anti-pure or racemic systems because the molecules have intrinsic chirality, generally speaking. So that for us was a really nice example of how we can put all of that together, but it's certainly not trivial, but really important to the sort of direct working with experiment there. So to move on to property prediction, so hopefully now I'm sort of showing 3 images of property prediction that would be very familiar to people from this community. So generally, if you have a solid state structure for your porous material, you can do void analysis, which I'm showing on the top left. In this case, this uses 0++, and the voids are coloured by whether or not they're accessible to a probe of a given size void representing a specific guest. In the middle, we can do sort of molecular dynamics simulations or something a bit fancier than that, but ultimately to get an insight into the different diffusion rates of different guests within that porous system so that you could predict that sort of separation capability. Or finally, you can do GCMC type simulations to reproduce what you would expect to get from absorption isotherms and to pick up to predict or understand from that capacities, but also potential for separations. So these are things I say if you have a solid state porous structure, but if given we had the aspiration to be sort of predictive and looking at hypothetical systems for these porous molecular systems, then we won't have the solid state structure very easily to hand. As I explained, if we're screening thousands of systems, it's completely not practical for us to also predict their solid state crystal structure too, because those simulations are too expensive. But at this point, what we thought to do was to take advantage of the fact that the systems are quite relatively unique within porous materials in that they are intrinsically modular and molecular. So here I've got a schematic trying to represent that. So here I've got the typical sort of tetrahedral symmetry cage. It's got 4 different cavity windows shown on the left. And then on the right is a very typical packing mode that you see, where each of these windows line up. So the 2 cages line up window to window next to each other, and therefore you get a diamondoid pore network. What I'm about to say next would apply whether or not you have a diamondoid pore network, but the key point that we identified is that if you really analyze the individual molecule and its structure, its void sizes, its cavity, etc., then you are actually analyzing all the key features that you have in the solid state in this case. Because if you had a guest diffusing through that diamondoid pore network, then the sort of key potential rate limiting steps are sort of the diffusing in and out of the window, and then also diffusing around the corner of the center of the cage. Much like getting sofa around a sort of circular set of stairs or something, you've got to do that awkward turn. But you could sort of predict how sorbates were going to behave within any hypothetical system by doing a lot of analysis at the molecular level. And so what we did was to develop software that would help us to do that. So the first thing Martin, who was an early PhD student in my group, did was to develop PiWindow. So this is software written in Python, and it's open source, it's there on GitHub. And what PiWindow does using Python is to, for a given cage molecule, determine the center of the cavity size. It also determines the number of windows. Here they're all colored differently, and what the size of those windows are. And something that's critical for us is that it's computationally cheap enough that we can run that analysis over a molecular dynamics simulation. Because while we're simplifying it by looking at the molecular level, we know it's really important to consider the flexibility and the breathing motions of that host porous material. But if we run a molecular dynamics simulation on a single molecule, we can get what many people call a Paul limiting envelope. So basically a histogram of the different void sizes that that host molecule can adapt. When you factor in sort of thermal breathing effects, and also we can do it with guests present, so you can see sort of cooperative behavior where guests might open up pores more as well. And surprisingly, we found that actually the packing wasn't having as strong an influence on all those sort of things like Paul limiting envelope for the cases that we looked at as we expected, and therefore this sort of approximation held quite well. But of course, that dynamics consideration is really important because a lot of the early systems, in fact, the first group to report this cage molecule, one of the classic cage molecules, actually wrote in their paper that this system the cavity is too small to host any guest, like any gas at all. Whereas in reality, that exact same system can even host a xylene molecule within it, but it's just all about that flexibility. But that group had just looked at the crystal structure and seen that, you know, the windows were too small for hydrogen and nitrogen, and assumed that it was impossible to get a guest in. But in fact, there's quite considerable breathing motion that means that you can. Right, so we have PiWindow for analysis of the sort of structure, and we took this forward with looking for xenon-krypton separation candidates from previously reported porous molecular systems that were in the literature. We took about 200 of them from the Cambridge Structure Database and revisited them just by doing the analysis on the single molecule level for xenon-krypton separation, by doing that structural size analysis, by doing DFT calculations, looking at the binding energies of the xenon-krypton, and also by doing metadynamic simulations that gave us insight into the barriers for removing xenon and krypton through the window of the cage. So it would be important for a sort of kinetic-based separation. And that identified a new system that was potentially promising, the one shown on the right, the Norio water wheel. And our experimental collaborators then synthesized it and tested it for xenon-krypton separation and found that indeed it did have the performance we were expecting. And we've done similar based approaches, also funded by industry, looking at separations of hydrocarbons and xylenes as well. So that's a sort of introduction into what we were trying to achieve. And obviously, as I said, we always wanted to be predictive and to be getting ahead by exploring new chemical space. And a key thing for us to be able to do that once we had this ability to do the structure and property prediction is that we needed to automate a lot of this process in terms of the building models and assessing them, so that we could go up to large scales of hundreds of thousands of systems inspected. So the key thing here was our SDK, or Supermolecular Toolkit, developed by talented student Lucas Tocani when he was doing his PhD in my group. So what this does is you can feed it precursors, generally organic precursors, although it can now deal with some metals. And we have functions within that code that allow you to assemble into different types of cage topology or different systems like linear polymers, small molecules made up from different units, COFs and so on. And you can then analyze their structural properties, like by connecting up to PiWindow, or we can analyze electronic properties, but I guess of less interest to this community. But again, this software is written in Python and is open source. You can see the GitHub page there, and I'll be showing that again later. And then this is an example of, we've been continually extending this, I'll show later, and we can now do a variety of different systems. So this is examples of structures that we can, in theory, build within SDK. Now, just having the ability to automatically assemble thousands of possibilities isn't enough when you've got enormous potential chemical spaces to explore. So we also extended SDK with Lucas and also Enrico to include an evolutionary algorithm. So this is a way to sort of efficiently search the space. So I'll just show you two key features of that. We're trying to mimic sort of survival of the fittest in nature and how that's working with evolution. So we need mutation and crossover functions. This doesn't have to be done on porous systems, but here I'm showing an example of, say you had a porous cube. A mutation is swapping in one of your building blocks for a new one. You can do it randomly or not completely randomly, depending on what you're doing. So here is swapping green for orange, or at the bottom you can do crossover, where you switch the precursors of two parent individuals to get two new offspring. And the other key thing that you need within an evolutionary algorithm is some sort of fitness function that you're seeking to optimize. This equation at the top is not to be taken too literally, but just to get the point that we can weight a variety of features that we want to see in our final material or porous molecule. So for instance, we first ran it when we were doing very simple calculations on porous cages, that we just were targeting a specific pore size, and there was penalties for systems that were far away from that. And then we also do a symmetry analysis as a way to sort of check for strain within the systems. So here is an example to sort of show how this works. I think it's interesting to sort of, or it's useful to help understand how the evolutionary algorithm works. And in this case, this was a particular case study where we were looking at C60 binders. So we were trying to find a porous molecule that could do a good job of absorbing C60. And what I'm showing here is the top candidate at any given generation. So there'll be, say, 64 candidates in a generation, but I'm showing you the best one. And then what's numbered is the generation number. So at the initial guess, that's largely random, it formed a cage that really isn't big enough for C60. By the next generation, generation 1, the cage has got a bit larger. So, and then it sort of slightly overdid that. By 3, it's got too large. And then it sort of settles down thereafter in the generations on cage sizes that are much more sensible for hosting a C60. And so instead what you're seeing is that cages are getting selected for where there are switches of the precursors that will help to strengthen the binding energy for the C60. And then we would run it multiple times to sample different outcomes. And here is then our top 20 systems that were predicted with a 4+6 topology to strongly bind C60. And yeah, this is just to emphasize that SDK is something that we are continually still expanding within my groups, expanding to look at new types of systems, like adding in new sort of 2D COFs, this sort of thing. We've also done coordination cages, retaxanes, a whole variety of things. And we have, as well as the GitHub where the source code is, there are some YouTube tutorials that Andrew Tarzia, who's in my group, wrote to help people get started. And there's a Discord channel that you can join if you want to get help from some of the group members that use and develop SDK, if you want to extend it to a new system that yourself or some host guest complex or some other network system. And what I thought I'd very briefly mention is an example of particularly Andrew Tarzia's work in my group, where he extended SDK to working on coordination cages. And on the left-hand side, I'm just showing a snippet of the code within SDK written in Python that would help you assemble that tetrahedral coordination cage shown on the left. And these are, for us, quite sort of challenging systems potentially to model. As I said, we try to stay away from the metals because when we're doing screening, we don't really want to have to use DFT calculations to always get accurate structures. So what Andrew had to work on for a long time is finding methods that were cheap to sort of get sensible structures that we could use within SDK. But here are several images of different cage systems that SDK automatically built for him once he got that working. And I'm not going to go into much detail on this, but Andrew did a variety of different things looking at coordination cages with SDK. And this was an example where we were working with Jamie Lewis, who was previously a colleague here at Imperial, but is now at the University of Birmingham. And so Jamie was interested in looking at sort of low symmetry systems where you had linker molecules that were of lower symmetry, so had different end groups on either side, and then it gets really additionally complicated because you've got lots of possible different isomers. And we did a screening of 60 different systems and each of their 4 possible isomers, and try to assist the synthesis by comparing the energetics of the different outcomes and directing, therefore, Jamie to the systems where there was a strong preference for one isomer, which was what he wanted from his perspective. And that did work quite well. He then did go in the lab and make several of those systems that we had predicted for him. Although one of the systems we made we predicted he wasn't able to make, or it didn't make a clean single isomer. So I think just in terms of being honest, we're not saying what we can do is always perfect, but by getting really deep understanding and working closely with experimentalists, what we're always aiming at is to, you know, to be really making a difference and to accelerate the experimental discovery, and certainly to be much better than sort of what you're able to guess from a lot of years of sort of hard chemical experience in the lab. And so we're continuing to work on these sort of things, but as well as sort of more helping with predicting the structures and which of them can form, I think an interesting point is that you could also look through your hundreds of candidates of different possible molecules and look for features and properties you want. And so some simple analysis that Andrew did for some of these systems, where he could pick out different systems and rank them by the degree of anisotropy of the cavity that the low symmetry cages had. So if that was something you're interested in for binding a low symmetry guest. Right, so that was my sort of stuff on porous molecular materials, but I obviously did promise to get to membranes. And I guess this brings us to sort of a different tranche of the work we do in the group, which is modelling amorphous porous systems. And I guess, to be frank, started at looking at this much more with the interest in the sort of difficult structure prediction problem, and the fact that because you don't have crystal structures, modelling can really help, because if you can get realistic amorphous structures, then you can help with the sort of microscopic understanding that can be much more difficult for amorphous systems compared to crystalline ones. So yeah, and then we explored a lot sort of the influence of disorder and defects. So for amorphous moths, I guess we are brought into this topic by Tom Bennett, that does a lot experimentally on amorphous moths at the University of Cambridge. And he tries to make amorphous moths via a variety of different routes, as do others, sort of melt quenching, ball milling, or temperature or pressure induced amorphization. Although you can also do it via direct synthesis. So you're potentially accessing different bits of the amorphous phase space, I guess is the point. So I thought I'd pick out one particular case that we've done with them. So Tom and Adam, who was a PhD student with him at the time. So it's this iron FeBTC system, which contains the same building blocks as Mil 100 and also basalite F300, which is used commercially as a catalyst. And the sort of structure of Mil 100 is known, it's crystalline, but for the other systems, this was much less known, and they all have different surface areas and porosity. And so they're made via different synthetic routes. But as I say, they have the same underlying building units. And what I'm showing on the right-hand side is that you have different sort of order at different length scales. At the short range, you have the trimer unit that has 3 ions, and then you can build that up to a tetrahedron with the linkers, and then you get at long range order more of the building units combining together. And this tiny plot here is just to say, I'm about to sort of talk you through some of the simulations that we've done here. But actually, we were really surprised. We used an approach that I'll explain in a moment to sort of try and predict amorphous structures. And we're really surprised when we compared to a lot of the scattering data, how good a job we were doing at reproducing those. So I think the method by which we build them is relatively simplistic. So I don't think the model's perfect, but actually it's surprising the agreement we have. And so I think some of the insight that we get is pretty useful. So this was work in particular done with Irena, who's just finishing her PhD in my group now. So what we use is some we're using some software called Polymatic, developed by Corey Kalina's group in the US, and I have a reference for that on a later slide. But essentially, Polymatic works by doing a random packing of your monomers, and then doing loads and loads of rounds of bond formation to sort of polymerize the structure, and then you do sort of molecular dynamics procedures to anneal the structure at the end, and you make sure that you've capped any unreacted end groups. So in Irena's case, what was interesting about what she could do with the sort of potential FeBTC structures is that we, at the modeling level, have a control of the degree of disorder and also the degree of defects. So if I explain how we do that, like in terms of the degree of order, we can put in different sized building units. So as you've got on the left, we can just put in the relatively small unit, or we can be building up to larger secondary building units like the tetrahedron. So that controls the degree of short, medium range order based on how many of those we sort of throw in at the initial stage. And then we can control the level of polymerization, so how many bonds are formed, and therefore how many remaining defects that you get, where the defects are sort of unreacted end groups. And so then that allows us to do the comparison that this slide, I think, shows very nicely, whereby we have 9 different systems that have different degrees of order and defects. So on the left-hand side, we've got 3 different simulations, somewhere we only have the really small building unit. At the bottom, we have the large building unit, and then in the middle, we have a mixture. And then going along the x-axis, we have different levels of defects. And then what these images show is just a slice of the structures that we then model, the amorphous moth structures we get in the end. Tetrahedrons are where you've got an existent where you end up with a tetrahedron. You can see that largely these only occur when you preform them in our case. And then the colours are showing you how many unreacted end groups, so how many defects you've got in different areas. So we can see the various trends we're getting there by how we've controlled the simulation. And we could go back and compare this to experimental PDFs and scattering data by Adam and David Keane, and see the different trends that you get when you can change defects and when you change the level of order. So we can explore trends in different directions and compare that to the experimental patterns that they're getting when they change things in the synthetic routes. And then we can also go, okay, but does that tell us anything about the change in the porosity? And I think this sort of all intuitively makes sense in that we see that as you increase the amount of order, or you put in larger secondary building units, you're tending to trend towards larger cavity sizes. It's shown the top in the sort of orange colours, whereas changing the degree of defects is only having a very minimal size on the porosity, if we're just looking at sort of pore size distributions, although of course changing the level of defects will have a much stronger influence on other properties beyond porosity. And so in the green, we sort of go across that graph where we're changing both order and defects at the same time. But really now to get to the polymer membranes. So where we've done work in this area has mostly been looking at polymers of intrinsic microporosity. So these originally developed in particular by Neil McEwen at the University of Edinburgh. So perhaps in this crowd, people are already familiar with these, but the basic idea is you form these polymer chains that are quite contorted and are unable to pack efficiently. And so you end up with the sort of porosity that we see on the right-hand side. So we use a similar approach, and here is that reference at the bottom for polymatic, which we adapted to our purposes for both the moth and polymers here. But you do the same procedure. So you take the monomers of the PIM you're making, load them into a very large box at a low density. Then you do the polymerization procedure where you'll form hundreds of bonds and do lots of annealing to allow the structure to relax. Then when you're finished with the polymerization, you'll sort of do sensible chemical saturation of any of the unreacted end groups. And then you do a very long annealing procedure at different temperatures and pressures and so on to try to get sort of realistic structures. And that sort of typically makes the structure much more dense than you get at the end of the polymerization. And then obviously you can do lots of analysis of the structural properties of the system that you get there. So this is an example of 3 different systems that we modeled very early on using this approach. So 3 systems all made with a triacyl chloride shown at the top, but different reaction partners, those 3 organic molecules shown at the top. And so then in the middle, we just have a sort of normal display of the polymer models that you get with the voids shown in blue. And then at the bottom, the different voids are shown where they are coloured by the void size. And so what we find is very useful is that you can sort of compare the trends. And in this case, very subtle differences in the pore sizes, but the trends of those differences all sort of tied up with the actual sort of separation performance of these membranes. So it's a relatively sort of simple way of looking at this and just doing the void analysis, but it's already very insightful. And it's what we found ourselves doing quite a lot. And often, I think it's this point of where, compared to having a crystalline structure, you're a little bit more in the dark about sort of trying to rationalize structure property relationships. And I think the amorphous models can be really helpful for that. Unfortunately, what we often manage to do is to sort of ruin someone's hypothesis, or have a hypothesis for why something's working. Maybe it's hydrogen bonding network or something like this, and we'll analyze our models to that and then say, yeah, no, it's definitely not that that's causing the problem. But of course, all of this insight is still useful, even if we're generally sort of breaking hypotheses apart. And then hopefully I've got a couple of minutes to finish on something else. So that's a sort of direct modeling of polymer membranes. But I haven't spoke about sort of another theme of research in my group, where we have quite a lot of people looking at the use of machine learning or other techniques along the lines of artificial intelligence, to see where we've got problems that can't really be solved by molecular simulation, but potentially can be solved using machine learning. But and this is a problem in a lot of areas of chemistry or materials, if you have the necessary data in order to sort of train some sort of machine learning model. And in particular, so something that he did a few different projects on different types of materials where he was using machine learning, but within polymer membranes, he found this database from the Membrane Society of Australasia that people here are quite familiar with, where there's lots of polymer gas separation data that's there. And for a variety of different polymers. But if you look, and then so this plot shows that, of course, people don't measure every single guest within every polymer system that they measure. So there was a lot of what we could think of as missing values in that separation database, where a polymer has been tested for, say, hydrogen and oxygen, but not for, I don't know, helium or whatever. So you don't have a full set of data. And in theory, that means you may have missed opportunities for performance or separation capability, because you're not able to collect every single guest at a time. So the idea was, can you use some of the data that's in the database to fill in those missing points? And so that's using a type of machine learning called imputation, so imputation is just a word for sort of filling in. And so we were imputing the database to those missing values, and in particular using what's called mice algorithm, so imputation by chained equations. And we tried a few different flavors of different algorithms for that, and exactly which one works best sort of depends on exactly what you're doing, and you can read about that in the paper. But so say you have, this is a sort of flow chart just showing how it works. You have the missing values, you initialize some values, and then you try to build a model that predicts one specific guest at a time. And then you move on to the next guest, and you keep iterating around until you've converged, so you're not getting any performance. And I think I don't have the plot here, but generally it did quite a good job. You can obviously test by sort of removing some of the data and seeing how you'd reproduce it. And so it could do a pretty good job of reproducing the data. And so therefore we could try to look back and say, okay, can we now, with our filled in data points, identify promising systems? So if we look at the two plots here, so I guess this crowd is familiar with what we're trying to do. We're trying to break these sort of trade-offs between selectivity and permeability to find better systems. And so in these plots, so the left hand is for CO2 against methane and the other is for CO2 against nitrogen. All of these points are new ones where we can now do this comparison because we've filled in the missing points. So the blue points are where the CO2 was missing, the red is where the methane was missing, and the black is where both were missing. And so on what you can see labeled is that we found that there was this polymer that looked really promising, this PIM that's at the bottom. But when we went back, we saw that this had been reported previously in the literature, and it did indeed have the sort of performance that we had predicted, that it would have a very good performance. But it had been done, but it hadn't made its way into the database. So at least that was reassuring that this sort of approach could work to identify systems, although I guess it shows the sort of challenge that we have ahead of us for trying to use more things like machine learning and trying to really get all the data sort of curated in one place to take full advantages of this sort of approaches and technology. So in terms of how we view that this sort of thing could be applied is that, yes, you have an incomplete database and you could fill it in by this imputation. As I just sort of showed, you could use this to reanalyze existing data and identify promising systems where, for whatever reason, the full set of data or properties wasn't collected at the time. And now we can identify which to focus efforts on. But I think the other way we could envisage using this is to imagine that you don't necessarily need to immediately, for a new system, collect all of the data, but say you collect your permeability or whatever for CO2, that just knowing that, you have a machine learning model that then predicts lots of separation performances, so for instance, CO2 against nitrogen or also against CH4 here. But you didn't need to do that full set of experiments to know which systems were most promising for what application. And I picked that example there of CO2 because we found in particular that there were certain guests or gases that were particularly good to use in models with low data, sparse data. So we'd use different algorithms for where we had sparse data. So for instance, you had one guest and then you were trying to predict 5, and you might use a different algorithm if you already have 5 guests and you're just trying to predict a missing one, because that's sort of an easier problem. But there were certain guests like CO2 that were really indicative, and then certain ones like CH4, which were less good indicators. But I think it's speaking with experimental colleagues, if I've got that the right way around, that also makes sense in terms of maybe our knowledge of where there might be noisier data that's in the literature, and also sort of physically as well, in terms of which ones are most likely to be useful for helping predict the others via the machine learning model. And yeah, hopefully, although it was a whistle-stop tour, it's still covered, or it still at least was clear about some of the various areas that we're working on within porous materials and the different types of techniques and approaches and focuses that we have. And so it just remains for me to thank my group and my collaborators. Hopefully I've mentioned the relevant people at the right time, and also the funding bodies at the bottom for funding. Thank you.
Thank you very much, Kim, for your talk. We have a few questions. If you're happy to get into those straight away.
Yeah, sure.
Oh, you can leave the slides up. That's okay.
Okay, that's just so I can see what's going on
there. Yeah. Earlier on in the presentation, or the first part where you were talking about the kind of materials screening, do you have any kind of pre-screening or rules or bounds that kind of limit the space that you search in, just to make your life easier in terms of computational time?
Yeah, so it really sort of depends what we're doing, and this is why it's so great to sort of work with experimental collaborators, because we will understand from them maybe what their limits are first, or we've actually gone to a lot of efforts and work that I haven't spoken to here, is to try and make the step of it can be quite easy for us to predict systems, but often our experimental collaborators look at all of them and say, basically almost all of them, maybe 95%, like these aren't possible to synthesize. Maybe the synthesis of the precursors is dangerous or really expensive. So we've done a lot of bolting on sort of things that can score the synthesizability for us to try and help with that. But I think there isn't a simple answer to that, because it will depend the problem we're working on, what exact property we are, the discussion with experimental collaborators. The idea of using the evolutionary algorithm was to sort of efficiently search the really large search space, because I think whilst we talk to experimental colleagues to try and make sure that there's a chance that we can synthesize things, I think something that is important to leave as a possibility with the computational modeling is that you could throw up a suggestion that the experimental researcher never would have tried or thought of, so that you are really doing something novel, rather than if we predict a load of things and then it turns out to be the things that somebody would obviously have already tried, if that makes sense. So it's a bit of a balance about trying to be really exploratory versus being safe to increase the chance that you can make it.
Yeah, that makes sense. And I guess that highlights a good kind of opportunities for like relationships and collaboration with those two like areas of research. But something that you mentioned in your answer kind of leads very well to kind of the next category of questions that have come up, which is the synthesizability aspect. Because when you were talking, when you had that slide, it kind of reminded me of the theoretical zeolite structures versus actually synthesizable zeolite structures. And you touched on that in your answer before, but do you? Yeah, so do you? I guess you answered, but do you have a method to kind of integrate that feedback into solutions? And another question is, have you noticed any trends from this that allow you to kind of predict why certain types of materials fail when they're trying to be synthesized or anything like that?
Mm-hmm. Yeah, so this is a really complicated problem. I actually sort of deliberately took out, I normally have quite a few slides on this, written some reviews recently, but I guess I assumed people were more interested in the porous stuff. But for me, a lot of this, it's really nice that you touched upon that point with the zeolites and so on, because a lot of my early work of what I did with these organic molecules was really inspired by all that earlier work on zeolites and comparing all the hypothetical polymorphs. And I guess I was exposed to during my PhD, and that I find really interesting because there's different elements to that of, comparing the relative energies. Is it thermodynamics, but also do you need to be able to, some things in theory should be possible to make, but maybe you needed to find the right template to guide that synthesis. And so it's much more complex than just the thermodynamics at the end.
Right, when you have the organic,
the sort of crystal organic polymorphs as well. But for us, for these, so for instance, to take the organic cages, there's all sorts of different levels to that synthesizability part. There's the can you make the precursors, which is where we built a machine learning model that basically is trained to think like our experimental chemist, in the sense that we got her to rank 10,000 molecules, and then we just built a machine learning model that could predict her ranking of basically yes, no, I will make that, so that we can do that within a fraction of a second and add that as a bolt on to what we're doing. But it's not just that, it's will the right molecule form, and that for me is really difficult because I think that solvent and things like that are having a lot of influence, and that's quite hard to sort of adequately capture from an experimental perspective. So I think from a computational perspective, rather, by doing molecular simulations. So in a sense, I've sort of thought, okay, maybe I will not do it that route and do it via different routes in the future. But then you've also got, does it remain soluble? So that, for instance, if you're making a membrane, can you cast it there? So there's loads of different levels, and it's specific to each material class, I guess. But yeah, this is a problem that we're really interested in. And I think it often requires working in an area for quite a long time and lots of discussions with the experimental researchers, so that we can pick up, okay, what descriptors might we be able to use in our model that can help us increase our chances. But I think that's going to be like a lifetime's work, maybe, to just do better at that.
Yeah, no, that sounds great. Obviously, yeah, there are obviously so many factors that are difficult to computationally reproduce, right? Which I guess is the limiting factor.
Yeah, and often there isn't that much experimental data either, so you don't know what's going on. Like, you're just looking at a couple of cases, and like solvents, I'm pretty sure solvents are a big factor, but there's, like in that particular system, there's only a couple of experimental reports that really show you what's happening with solvents. Because I guess that's another interesting point in thinking of data and how you could use more machine learning, is that all the failures are not there in the literature. And so we only can see the things that worked, and that's not really the full picture. It's like with the shape persistency, if 95% of things collapse. But in the literature at the moment, really, you'd only have the cases that worked. So yeah, trying to get
you have the bias kind of inherent to that. There's a couple more questions. One's from Nick Wilkins, which tied in pretty well with something that you mentioned just now about the machine learning kind of aspects. So how computationally difficult is this work? Like, is it a laptop scale kind of thing, or is it a computing cluster kind of thing? Could you give us some indication on that side of the story?
Yeah. So of course, again, the answer is a bit tricky because it depends. But what we're doing things, I guess, from the scale of like where we've built the machine learning models, which is including a link, but we have a website where people can then just use our website and sort of drop in a smiles code for a precursor to get some of our predictions. So that then requires nothing other than a browser, so people don't even need to be able to code. And then a lot of what we're doing is at the force field level, or, we've worked hard to try and not have to use DFT by finding methods that sort of in between force fields and DFT. In that case, sometimes we might run it locally if we've got a good workstation, with a few cores, but we're probably generally using the university computing cluster, so that we can be running things on, maybe some hundreds of cores, but not loads. And then sometimes we'll use like the UK supercomputer, Archer 2 as well for it, but that's really safe when we're doing lots of DFT calculations. So like not really on a desktop, but sort of not insane amounts of computing power either, I guess.
Right, like a kind of university accessible kind of level of computing power.
Yeah. And then maybe if you're just using SDK to sort of build structures, the idea is probably that can be done more locally on someone's own computer if you were just doing that. But it's like if you're trying to do the property calculations, then it's likely you're using a university cluster. Yeah.
Cool, thank you. Well, we're also happy to share any links to the community as well after the talk if you want to provide them to us. So that's not a problem. And one last question from towards the end of the talk on the kind of amorphous modeling and the polymer modeling. Is there any limitation or effect of, you mentioned the box size.
Yeah.
Right, so I guess the question is how small, what's the minimum size of the box size, or is there a relationship between the minimum size you need before it begins to impact the results that you see?
Yeah, so we tend to do, it's quite surprisingly small, I think, and I think people that are used to thinking of membrane scale on it, it's surprisingly small. It's sort of our box, typically about 70 angstrom, we find works fine, in that we have tested that if you go bigger, you're not really seeing changes in the properties that we're analysing. So 70 angstrom is obviously quite small compared to, I guess, the scales people are thinking of.
Yeah, that's pretty surprising, at least to me, who doesn't really know much about
that area. But if you
area.
we're just trying to sort of a snapshot of the bulk rather than, we're not trying to represent the full.
Right, but that is good news as well, ultimately. In
the
end.
But that's still, so it makes that sort of computationally quite accessible, but it's not trivial. That certainly we'd be doing on a computer cluster. But maybe we can do the whole process for a model in a week or two that way. But it quickly, because of how it scales, would get really expensive if you started to go larger. But yeah, we've checked for what we're doing, and we're careful to check that it's not an effective box size. Like we're not getting, what we're getting is converged for box size.
Excellent. Thank you very much. Thanks again for your talk. And also like to thank everyone joining the session today. And we hope that everyone enjoyed it. The next IAS webinar will be in early March, and details of that will follow soon. I'm now going to hand over to Nick Wilkins to wrap everything up.
Thank you, Dr. Jelfs, for your presentation From Membranes to Molecules, Computational Discovery of Porous Materials. We also thank all of the attendees for joining this webinar and hope that it was both educational and enjoyable. An edited version of this webinar will be posted on the IAS YouTube channel and Bilibili with an announcement on the IAS Twitter feed. The hope is that the work discussed today will be a useful resource for the adsorption science community in the future. The next confirmed IAS webinar will be given by Jacek Jagiello on March 8th at 2 p.m. UTC. Announcements regarding the next webinar and other IAS online events will be posted on our Twitter feed and through the mailing lists. With that, we thank you for joining us and hope that you will join us again soon.